So, students, today we will talk about embryogenesis of urogenital system. It's embryogenesis of urinary and female and male reproductive systems. They have common sources of origin, and today I'll talk about their development together, about development of two systems. So we'll begin from embryogenesis of urinary system, and then we'll talk about development of reproductive systems, female and male, because they have common origin and common sources of development. So here we can see development of urinary system and progenitors of urinary system, and let's look what are the embryonic structures which give rise to the urinary and reproductive systems. So here you can see transverse section of the embryo. It has ectoderm. Ectodermal structures are shown in blue color. It's dermal ectoderm, it's neuroectoderm, it's nervous tube or neural tube which give rise to nervous system. It's endoderm, it's shown in yellow. It will give rise to the epithelium of digestive and respiratory systems, and between them structures shown in different shades of pink are structures of mesoderm, and it's notochord. And mesoderm, it has differentiated part, here we can see it, and undifferentiated part, here we can see pale pink. Filler, it's undifferentiated mesoderm, it's called mesenchyme, it's embryonic connective tissue. And differentiated mesoderm, it has parexial part or medial part. There are somites on two sides of embryo. Next are intermediate mesoderm. Here we can see it. Intermediate mesoderm also is called nephrotome or nephrogonotome. And lateral mesoderm, here we can see it. It's called splanchnotom. And between two layers of lateral mesoderm, we can see cavity which is called coelom. So here we can see coelomic cavity. And there are two layers of splanchnotom. They give rise to serous membranes such as pleura, pericardium, and peritoneum. So here we can see those two layers. And today we are interested in the nephrotomes and their development because they are giving rise to urinary and reproductive systems. So here we can see next stage of their development. Here we can see that nephrotomes, intermediate mesoderm, they are separated from parexial mesoderm, from somites, and from lateral mesoderm, from splanchnotomes. And here we can see those nephrotomes. And note this splanchnotom, it will give rise to mesothelium of pleura, pericardium, and peritoneum. The splanchnotom also is involved in the development of urinary and especially reproductive systems. So here we can see two nephrotomes on two sides of embryo, and nephrotomes, they continue their development. Here we can see longitudinal section of embryo. It's simple picture which shows us ectoderm. Here we can see dermal ectoderm. Its anterior part, its embryonic oral cavity, its embryonic anus. There are ectodermal structures and its covering of the umbilical cord. Inside the embryo, we can see yellow endoderm. Here we can see it. It forms esophagus, stomach, and intestines. And also here we can see vitelline duct, which provides connection between endodermal gastrointestinal tract, its primitive gastrointestinal tract, and yolk sac. And between them, we can see mesoderm. And in this place, we can see nephrotome. It has shape of the cord. Here we can see this cord of cells, and they have undifferentiated structure. So it's common cell mass without division into any structures. We call this splanchnotom. And let's look what will, we call this nephrotome. And let's look what will happen to nephrotome on the next stage of development. Here we can see embryo will rotate this embryo, its head, its bottom, its body of embryo, and in head and neck. Also there is a nephrotome, so germs of kidneys, they are present in head and neck region. And this part, it's the earliest stage of kidney development. It's called pronephros. Pro, it's before, nephros, it means kidneys. So the earliest stage of the kidney development, it's called pronephros. Next stage, it's mesonephros, and mesonephros is developed in the thoracal and abdominal region of embryo. Here we can see mesonephros. It's the first stage of kidney which is functioning. So we call this also primary kidney. It's mesonephros and it's located in the body. But pronephros and mesonephros later they will disappear and they aren't functioning after birth. And the last final stage, it's metanephros. It's located in the pelvic region near the bottom of embryo. It's pelvic region. It's metanephros and definitive kidneys, which are known as kidneys after birth, they develop from metanephros. So it's the last stage, and germs of kidneys, they are developed in the pelvic region of embryo. So the first stage, it's pronephros. It's embryonic rudiment, it's not functioning. Next, it's mesonephros. It develops in the body and thoracal and abdominal regions of embryo. It's the first kidney which is functioning, so it's called primary kidney. And metanephros, it's also called secondary kidney or definitive kidney. It's called metanephros. And after birth, only metanephros is functioning. So let's look what is going on in this common cell mass and how it gives rise to pro, meso, and metanephros. So here we can see this nephrotome, and this common cell mass begins segmentation. It's the division into segments. This common cell mass is divided into groups of cells. They are called segments. And here we can see this segmented structure. This segmentation begins from head and neck region. And here we can see its gastrointestinal tract, its intestinal tube, and on two sides of the intestinal tube, we can see two nephrotomes from right and left sides, and in head and neck region, we can see segmentation of nephrotome, and we receive segments about 7 till 10 till 12 segments, they appear here, and it's the first stage of pronephros development. And after that, those segments, they form tubules which are growing to the center of the embryo. Here we can see those tubules. And later they will be fused and they form common ducts on two sides. Each pronephros has own duct. And this duct continuous growth into the thoracal and abdominal region of embryo. So we receive structure like brush or comb. There are segments and common duct. This duct is called mesonephric duct or Wolffian duct. And this duct, it plays key role in the development of the following stages of kidneys. And also this duct plays key role in the development of reproductive systems, especially male. So here we can see Wolffian duct and tubules of pronephros, which are opened in it. Tubules are opened in the cavity of body of embryo, so they have rudimental structure, but they aren't functioning. And later, after some weeks, they disappear. But mesonephric duct remains, and it will be functioning later. It's very important structure in the embryonic development of urinary system. So the main achievement of the pronephros is formation of mesonephric duct. And here we can see formation of this mesonephric duct or Wolffian duct. Two ducts are present on two sides of embryo. We can see their appearance on the transverse section. And here we can see tubules of pronephros which are opened in this duct. So here we can see structure of pronephros. And on the microscopic images, we can see tubules of pronephros. They aren't functioning, so simply epithelial tubules which are opened in the common duct. The next stage of the development is mesonephros. It's primary kidney because it's the first stage of kidney's development which is functioning, but it's functioning only during the embryogenesis, and after that its function is replaced by metanephros. So what is mesonephros? Let's look at its development. Here we can see pronephros, a result of pronephros development, and here we can see two unsegmented cell groups, cell mass, and it continues its segmentation. So segmentation occurs also in the thoracic and abdominal regions of embryo. Here we can see segmentation, and also those groups of cells, they are growing, they form tubules which are opened into the mesonephric duct. Here we can see them, and the situation is like development of pronephros. But there is one difference. These tubules, they form structures like nephrons. They form Bowman's capsules, cup-like structures, and blood vessels from aorta, they are growing to these nephron-like structures. So they are functioning like nephrons. And mesonephros is functioning stage of embryo development. And here we can see glomeruli and Bowman's capsules and tubules which are opened into the mesonephric duct. So there are structures like nephrons in the definitive kidneys, and those structures are called Malpighian bodies. Here we can see them. So it's mesonephric duct, tubules which are opened in it, and also glomeruli and capsules. Filtration takes place here, and urine goes through these tubules, and it's collected by the mesonephric duct. So mesonephros is functioning. And here we can see mesonephros. And here we can see its mesonephric duct or Wolffian duct, and there are small nephron-like structures which are present on two sides, but in definitive kidneys we know that nephrons are having special localization, and they are opened into the collected tubules and ducts, but in the embryogenesis there are two Wolffian ducts on two sides, and they collect filtrate from the Malpighian bodies of mesonephros. And here we can see mesonephros in human embryo. It's histological slide, and there are mesonephric structures, embryonic glomeruli and capsules. So Malpighian bodies are visible here. And here we can see growth of mesonephric duct, and nephrons, nephron-like structures, they appear here during the segmentation of nephrotome. And this duct achieves primitive digestive tube, it achieves primitive rectum, and there is common sinus for urinary and digestive systems, so it's common rectum and embryonic primitive urinary bladder, and we call this cloaca. So the collective part of digestive and urinary systems in embryogenesis is called cloaca, and some animals, they even after birth they have cloaca, but in human and in other mammalians, cloaca is divided later into the urinary part and digestive part. So it will be divided into rectum and urinary bladder. And here we can see mesonephros, and next stage is formation of metanephros or definitive kidney. Here we can see formation of the final structure of kidneys. So what will happen next? These ducts, they achieve the digestive tube, they are opened here, and this urine filtrate is opened into digestive tube. And we can see here that in pelvic region, nephrotome remains without segmentation, and we call this metanephrogenic tissue, which will form parenchyma of kidneys. And this mesonephric duct, it's Wolffian duct, and it forms two diverticuli, which are growing to their metanephrogenic tissue. So we'll label them with different shades. It's metanephrogenic tissue without segmentation, and there are buds of ureters. Here we can see growing ureters. They are divertically of mesonephric duct. And they are growing to the metanephrogenic tissue, and they are growing inside the metanephrogenic tissue, and they form urinary tract inside the kidneys. So we can see two sources of kidneys development. It's metanephrogenic tissue or metanephrogenic mesenchyme. It's nephrotome without segmentation, and also there are diverticuli of mesonephric or Wolffian duct. They form ureters and also urinary tract inside the kidneys. Here we can see structures of kidneys which develop from different sources. Pelvis and calyces and ureters, they develop from diverticule of mesonephric duct, and metanephrogenic tissue, it forms parenchyma of kidneys. And here we can see development. So it's mesonephric duct, it's opened into cloaca, common sinus of rectum and urinary bladder. And here we can see diverticule, it goes to kidney and it will be ureter. So here we can see bud, renal bud, and later it forms major and minor calyces of kidneys, and its first source of kidneys development. Here we can see minor calyces of kidneys. It's metanephrogenic tissue. It forms renal pyramids, and collecting tubules also are formed from metanephrogenic tissue. They are growing inside the renal pyramids. They form medullary rays inside the renal pyramids, and they also are source of development of kidneys. And here we can see development of the collecting tubules and ducts. And here we can see growing collecting tubule. It's located inside the kidney and it's surrounded by metanephrogenic tissue. It forms groups of cells, and later they will form tube-like structures with cavity, so something like bubble, and this bubble is elongated, and later it forms S-shaped body. It will be nephron. It's called S-shaped body, and later these tubules are fused, and they will form part of nephron renal tubules. And later blood vessels from mesenchyme. It's the third source of development of kidneys. Mesenchymal blood vessels, they grow inside this cup of S-shaped body, and they form glomerulus. And after that, this loop is rotated and it forms the nephron. So renal tubules and this capsule, they are developed from the metanephrogenic tissue, and connecting tubule and collecting tubule, they develop from diverticule of mesonephric duct. And here we can see development of the nephron. So blood vessels from mesenchyme, they grow inside the Bowman's capsule, and they form glomerulus. Bowman's capsule and renal tubules, they develop from metanephrogenic tissue, and connecting tubule, short tubule between nephron and collecting tubules and ducts. And the following urinary tract, there are calyces, minor and major, pelvis, and ureters. They develop from diverticule of mesonephric duct. And here we can see development of nephron. So it's mesenchyme, metanephric or metanephrogenic mesenchyme. It forms vesicles, and then they form S-shaped bodies. And cells are called renal progenitors, and also there are tubular progenitors which are growing from the Wolffian duct. And here we can see connection and formation of nephron. And here we can see metanephros in human embryo. It's 6 weeks of development. So when mesonephros is functioning, metanephros is already developed. Here we can see it. And there are numerous nephrons in the metanephric kidney, its final kidney. And later we can see here different developing tubules, renal bodies, and its metanephros in human embryo on week 12. And here we can see fetal kidney also developing nephrons, developing tubules, and till birth, kidneys they achieve their final development. And here we can see kidneys. So they are developed in the pelvic region. So they also are called pelvic kidneys. And sometimes they remain in pelvis, and it's also a mistake of embryogenesis. But usually in normal conditions, they should move in the abdominal region. Uh, Pronephros, it disappears during the early stages of embryogenesis, and it's not involved in the further development of embryo. Mesonephros keeps functioning, but later when mesonephros completes its development, mesonephros stops functioning, and later it's involved in the formation of the reproductive system. So these tubules are very important in the development of reproductive system, especially male. And this metanephros, it's the only functioning kidney stage development which is present after birth. So only metanephros will produce urine after birth, and previous stages they will not produce urine. And after the development in the pelvis, metanephric kidneys, they are elevated, they are going to the abdominal region. Ureters are growing, and kidneys also are coming upper, and also they are rotated, and they take their definitive position. So it's ascent of kidneys. And here we can see development of kidneys in the pelvic region, and after that they are moving and they receive their final position. And also they change their position, they are rotated on the 90 grades, and here we can see this position. And we can see elevation of kidneys, they are coming to the abdominal region, and they achieve their final position in the embryonic body. And here we can see it's the last stage of metanephros development, and kidneys are developed on this stage. And here we can see it's mesonephric duct or Wolffian duct, it's ureter, and it's cloaca. And later this cloaca and this region will be divided. Those structures, they will be divided, and they will form definitive structure of this urogenital and cloacal region and anal region. So here we can see development of embryo. It's ectoderm, it's endoderm, and you remember that ectoderm it also is involved in the formation of digestive system. Endoderm on early stages has blind ends, so it's digestive tube, but it has both blind ends, and how this embryo will eat and how this embryo will release the waste products. So ectoderm is involved in the formation of stomodome and proctodome, and also there is it forms stomodome invagination in the anterior part and proctodome invagination in the posterior part. And also here we can see process of endoderm, which is growing into the umbilical cord, and this process is called allantois. Here we can see it. It's very important embryonic structure which is involved in the formation of urinary bladder. And also allantois takes part in the growing of blood vessels. Blood vessels, they follow allantois from embryo, and they are growing inside the umbilical cord, and they achieve placenta. So they are following the allantois, and it's also function of allantois. And here we can see allantois process of the digestive tube in the lower part, and it's involved in the formation of urogenital region of embryo. So here we can see digestive tube, it's proctodome invagination of ectoderm, and they will communicate, this membrane will be broken. It's called cloacal membrane, and allantois is growing here, Here we can see allantois, it goes to the umbilical cord, and later it will be urinary bladder. So here we can see common cloaca, and later it will be divided into urinary bladder and rectum. And here we can see that this duct is opened into the urogenital sinus into allantois. It was opened into cloaca, now it's moving to allantois. And let's divide these colors. So this diverticule, it is ureter, developing ureter, and its mesonephric duct, and later they will be separated from each other. Here we can see that ureter is separated from the mesonephric duct, and it achieves the final position. Here we can see it. And mesonephric duct, it remains, and in male organism it will form ductus deferens. In female it will disappear. So here we can see final development of the ureter. It's opened into the allantois, into the urogenital sinus, and it's separated from the mesonephric duct. And here we can see that it's going upper, and mesonephric duct it's going lower, and later it will achieve urethra. And here we can see division of the urogenital sinus. Now it will be urinary bladder, and its rectum, which is opened through the proctodum to the outer environment. And here we can see formation of the urinary bladder and urethra. It was before, and it's after they were separated. So urinary system and digestive were separated during the embryogenesis. And here we can see that this duct in a female organism, it disappears, and later we will see what happens to this duct in male organism. And here we can see final development of the urinary bladder. It will be urethra, urinary bladder, and upper part of allantois is obliterated, so it was opened and now it's obliterated, and it will be called urachus. If you remember anatomy, there is plica umbilicalis media. It's midline of the inner wall of abdominal cavity, so inner abdominal wall, inner surface of the abdominal wall. And there is plica umbilicalis media, and it contains urachus. It's obliterated allantois. It's embryonic structure. And usually it's completely obliterated, but in some cases it remains open, and in those cases urine may be released from urinary bladder through the urachal duct into the umbilical region. So urine may be released through the umbilicus when this urachus is not obliterated. But it's mistake of embryogenesis, and in normal condition it's obliterated. So it remains in the organism in adulthood as a plica umbilicalis media. And here we can see division of cloaca into two, into urinary part and digestive part into rectum and urogenital sinus, and then urinary bladder. And we have discussed development of urinary system, and now let's look at embryogenesis of reproductive system. Those systems are very related, especially in their embryogenesis, and some structures of the urinary system, embryonic structures of urinary system, it's mesonephros, they are involved in the development of reproductive system. So let's look. Here we can see reproductive systems, and development of reproductive system. It has two stages. It's indifferent stage when we cannot distinguish boys and girls. So there are no differences between boy and girl. Is indifferent stage. And second stage is differentiated stage. And now we can distinguish girl and boy. So there are two ways of development, female and male. And those ways of development are determined by the genotype or karyotype. So when there is Y chromosome, there are genes which provide development in the male way. And if Y chromosome is absent and only X chromosomes are present, there is a female way of development. So if embryo has Turner syndrome, X0 karyotype, when only one X chromosome is present. This embryo has female way of development because Y chromosome is absent. So only one X chromosome is enough to provide female way of development, but if Y chromosome is present, male way of development will occur. So it's main key factor which determines the way of embryonic development, female and male. So here we can see in different and differentiated stages, and we will describe the development of reproductive systems for the gonads, ovaries, and testes. There are indifferent gonads and differentiated gonads, ovaries and testes, and also there is a reproductive tract. In female organisms, there are fallopian tubes, uterus, and vagina. In a male organism, it's seminiferous tract, different ductally efferentes, ductus epididymitis, ductus deferens, ejaculatorius, etc. And there are two reproductive tracts. They have common sources of development, and later we'll see their development. So here we can see pronephros, mesonephros, and metanephros. Metanephros is developing, it's functioning, and mesonephros stops development and it stops functioning. And now it will be involved in the formation of female and male reproductive systems. On this stage, embryonic gonads appear in the abdominal region. They appear on two sides of mesonephros, and they are indifferent gonads. So we cannot decide is it boy or girl. They have common early stages of development. And here we can see this primitive or primary embryonic gonads. They are located near the nephrotome. They form genital ridges. Here we can see splanchnotom, its cavity of splanchnotom, its coelom, and its parietal layer of splanchnotom. It will be parietal peritoneum, it's visceral, and there are genital ridges where two layers of splanchnotom are communicating. It's located here, and under this mesothelium embryonic, under this splanchnotom, there are genital ridges. They are shown in green shade, and they are located near the Wolffian duct. So they will be or ovaries or testes. Here we can see nephrotome. It's embryo, it's ectoderm, it's endoderm, it's notochord, neural tube, and here we can see genital or urogenital ridges. They are invaginations of nephrotome into the coelomic cavity, and they are covered by the coelomic epithelium, its splanchnotom, and here we can see two nephrotomes. And here we can see genital ridge, it's growing, so they will be or ovaries or testes. And the cells from mesenchyme of the yolk sac, they migrate through the blood vessels. They are called gonoblasts. They migrate and they are coming to the genital ridges. So they will form gametes. They will involved in the oogenesis in female organism or in the spermatogenesis in male organism. So gonoblasts, they are progenitors of ova and spermatozoa. And we can see that they are coming from the yolk sac and they migrate to the embryo. So they are coming from extraembryonic organ and they are coming to embryo. So they are some kind of aliens here, and they are located in the own tissues of the genital ridges. And here we can see migration of gonoblasts into the genital ridges. Gonoblasts, they migrate here through the umbilical cord from the yolk sac. So yolk sac, it provides this function. And they migrate in the genital ridge, and they are involved in the formation of gonads. And here we can see genital ridge. So it's invagination of mesenchyme into the coelom. it's a coelomic epithelium or it's embryonic mesothelium of the embryonic peritoneum. So embryonic peritoneum, it's parietal layer, its visceral layer, and here they communicate and they cover the genital ridge. Here we can see it. And this epithelium which covers the genital ridge, it's mesothelium. And you remember that ovaries and testes, they are covered by peritoneum, and this peritoneum is called germinal epithelium, especially in ovaries. So it's coelomic epithelium or splanchnotomes or peritoneum, embryonic peritoneum, which covers the ovaries and testes. So what will happen to this epithelium? It plays key role in the development of the ovaries and testes. It's growing and it forms invaginations in the mesenchyme in embryonic connective tissue. Here we can see cords, genital cords, which are growing inside the primitive gonad. Here we can see those cords, and they will form microenvironment for the development gametes, ova and spermatozoa. So this germinal epithelium forms Sertoli cells in testes and follicular cells in ovaries. Those cells, nurse cells or shaper cells, which provide microenvironment for developing ova and spermatozoa. And they develop from this germinal epithelium. And gonoblasts, they migrate here and they take their final placement in the genital ridges, in the genital cords. Here we can see those gonoblasts. So it's in different stage of embryonic development. Here we can see it, and here we can see in different gonads. So now we cannot distinguish between boy and girl. And let's look what will happen next. So in male way of development, we can see division of these genital cords into the testicular cords. Inside they contain gonoblasts, and there are cords of germinal epithelium. They are separated from the covering germinal epithelium. So this germinal epithelium which remains on the surface of testes, and it will be called germinal epithelium. Deeper, this mesenchyme will form tunica albuginea, and these cords, testicular cords, they will form convoluted seminiferous tubules, place where spermatozoa are developed. And they are formed by spermatogenic cells. Here we can see gonoblasts, and the basis of these cords is germinal epithelium, which came inside the testes. So here we can see those cords, testicular cords, and they will form convoluted seminiferous tubules. And germinal epithelium will form Sertoli cells, and gonoblasts, they will form spermatogenic cells. Here we can see those gonoblasts, and they will form spermatogenic cells. And here we can see testicular cords. They were completely separated from the superficial germinal epithelium. And here we can see the superficial modified peritoneum, which covers testes and also ovaries. It also gives rise, it's related with the basis of the testicular cords. It's related with Sertoli cells, which will form testicular cords. And here we can see transverse section of the convoluted seminiferous tubule. And here we can see Sertoli cells. They develop from the germinal epithelium, so they develop from splanchnotom, from coelomic epithelium. They come here from the surface. They form the basis, and they are shown in pink, dark pink color. It's germinal epithelium. And spermatogenic cells, they are shown in green shade. They develop from gonoblasts, so they have own source of development, and they fill the gaps between Sertoli cells. Sertoli cells, they form microenvironment for the developing gonoblasts, and gonoblasts, they migrate here, they take their placement, and they develop under influence of Sertoli cells. So Sertoli cells, they develop from this germinal epithelium is coelomic epithelium or modified embryonic peritoneum, and those cells migrate here. They form the basis of the cords. And inside there are gonoblasts, which develop into the spermatogenic cells. And those tubules and first of all testicular cords, they will form convoluted seminiferous tubules. They are located in mesenchyme embryonic connective tissue and mesenchymal cells. Some of them they form Leydig cells. Here we can see Leydig cells which produce testosterone. They develop from mesenchyme. So these cells, they are shown in red. They develop from mesenchyme, and they have third source of development. So there are three sources of development of testes. Gonoblasts, they give rise to spermatogenic cells. Germinal epithelium or splanchnotom, it gives rise to Sertoli cells. And mesenchyme, it gives rise to the Leydig cells. And myoid cells, fibrous layer, and other layers which are surrounding convoluted seminiferous tubules and also stroma of testes, they develop from mesenchyme. And here we can see testicular cords or testis cords, and here we can see that they change their shape, their appearance. Tunica albuginea, it develops from mesenchyme, from this mesenchyme, which is located under the germinal epithelium, which remains on the surface of testis. And also it forms, mesenchyme forms septa of testis, which separate testis into the lobules, and also intralobular connective tissue develops from mesenchyme. So connective tissue, usually it develops from mesenchyme, and in different organs, stroma, it's developed from mesenchyme, embryonic connective tissue. And also we can see that testis, it has rete testis and ductuli efferentes, ductus epididymitis follows them. And the sources of their development, it's mesonephric duct. And mesonephric duct gives rise to the seminiferous tract. So here we can see fetal testis, transverse sections of the seminiferous tubules, and there are developing Leydig cells between them. They develop from mesenchyme, and it's development of the parenchyma of testis. And later we'll talk about the development of the seminiferous tract and male reproductive tract, because other structures are involved in this formation. And now let's look at the female way of development. So female way of development of the gonads also has in different stage. When ovaries are similar to testes, also there are genital cords which are growing inside the mesenchyme, and gonoblasts, they migrate here and they are located in the genital cords. But in testes, there are testicular cords which are separated from the germinal epithelium. In female organism, those cords are growing deeper and they form islets. Here we can see islets containing gonoblasts, not the cords, not the elongated structures, but islets, groups or clusters of cells surrounded by the germinal epithelium, and later they will be divided into individual follicles. So inside each follicle, here we can see oocyte, primary oocyte. So gonoblasts, they form oogonia, which are proliferating inside the islets. Oogonia are proliferating, and then they enter in meiosis, and they become to be oocytes, primary oocytes. And those clusters of cells, they are divided into small follicles. This common cell mass is divided into small follicles. Each follicle contains only one cell, which develops from gonoblasts. And here we can see result of this. Here we can see individual follicles. So in the here we can see cluster of the gonoblasts surrounded by the germinal epithelial cells, and gonoblasts, they form oogonia which undergo mitotic divisions, numerous mitotic divisions, its proliferation, and then they enter in meiosis. They become to be primary oocytes, and each primary oocyte is surrounded by the germinal epithelium cells, and now they are called follicular cells. So germinal epithelial cells in testes, they form Sertoli cells. In ovaries, they form follicular cells. Here we can see them, and they develop from the germinal epithelium, and oocyte in prophase it enters in prophase of first meiotic division, and this division is stopped, and follicles are waiting for their further development after birth, after puberty. So here we can imagine how huge is the germ which is taken by the first meiotic division. And here we can see formation of the follicles. So here you can see follicles, oogonia, and then oocytes. They develop from gonoblasts. Gametes, they develop from gonoblasts. Both spermatozoa and ova, they develop from gonoblasts. Supporting or nurse cells, here follicular cells, they develop from the germinal epithelium, and germinal epithelium, which remains on the surface of the ovary, is called germinal epithelium. It's modified peritoneum, and its traditional name of this epithelium. That's why it's called germinal, because it in the embryogenesis it forms genital cords, and it forms nurse or supportive cells of the ovaries and testes. In ovaries, there are follicular cells which surround the ova, and they form their microenvironment. Cells which develop from the germinal epithelium, Sertoli cells from testes, and follicular cells from ovaries, they have common features. They develop from the germinal epithelium. They form microenvironment for developing gametes, ova or spermatozoa, and both are sensitive for FSH. Cells in the spermatogenic epithelium, Sertoli cells, they are sensitive for FSH, and follicular cells, they also are activated by FSH. So both follicular cells and Sertoli cells, they have common sources of development, and they are sensitive for FSH. And they have common function. They form microenvironment for developing gametes. And mesenchyme, it forms connective tissue stroma, which fills the space between the follicles, and also cells of theca. Theca appears later during big growth. Cells of theca, which are producing androgens, they develop from the connective tissue, and those theca cells, they produce androgens which are uptaken by the follicular cells, and they produce estrogens from them. So here we can see these following stages. And in testes, if you remember, androgens, it's testosterone, it's also produced by cells which develop from mesenchyme, Leydig cells, and this testosterone is uptaken by Sertoli cells, as well as androgens are uptaken by the follicular cells. And Sertoli cells, they produce androgen binding protein, and only this complex of testosterone and androgen binding protein can make influence on spermatogenic cells. So they are sensitive to the complex of testosterone and androgen binding protein. So here we can see also similarities in ovaries and testes. And here we can see fetal ovary. All follicles in the embryogenesis are primordial with squamous cells, follicular cells. They are flattened, and follicles, they remain primordial till the puberty. So in girls before the puberty, all the follicles are primordial, and when girl achieves puberty, some follicles are activated each month, and they enter in big growth, but in other follicles they remain to be primordial. So they exist in this condition during many years. And here we can see results of the development. It's male and female ways of development. In male way, we can see here testicular cords. In female, there are clusters of cells, and each cluster contains cell, its oocyte, primary oocyte, and both spermatogenic cells and oogenic cells, they develop from gonoblasts. Supportive cells, Sertoli cells and follicular cells, they develop from germinal epithelium, and remains of germinal epithelium, they cover testes or ovaries after their embryogenesis. And here we can see cells, gametes, they develop from gonoblasts, they are shown in green. Pink cells, they are supportive cells, Sertoli and follicular cells, and Leydig cells here and Teca cells here, they develop from mesenchyme and they produce androgens in both in female and male organisms. So let's make a summary. In testes and in the ovary, there are three sources of development. There are primordial germ cells. They form spermatogonia in testes and oogonia in ovaries. And later, spermatogonia will form spermatozoa, and oogonia, they will form ova. Next source is epithelium of genital ridge, coelomic or germinal epithelium. In testes, it forms tentacular cells or Sertoli cells. In ovaries, it forms follicular cells. And mesenchyme, in testes, it forms interstitial cells of Leydig, and in ovaries, it forms Teca cells. And also mesenchyme, it forms connective tissue stroma in testes and in the ovaries, and it forms tunica albuginea of testes and ovaries. So all connective tissue structures are formed by mesenchyme of the genital ridges. So here we can see sources of development, and we can see that testes and ovaries, they have common sources of development, and they have also differences in differentiation. In testes, there are genital testicular cords, and in ovaries there are clusters of cells which result in the formation of convoluted seminiferous tubules in testes and follicles in the ovaries. And here we can see localization of genital ridges. They are developed into the ovaries or testes, and they develop in the abdominal region. And here we can see a development of the gonads, places where they are developed. And now let's look at the development of the reproductive tract, female and male. There are some similarities, and also there are two stages in different stage when we can distinguish boy and girl, and differentiated stage when there are some differences in the structure of the reproductive tracts. So we will left only the Wolffian duct. Here we can see this mesonephric duct. Pronephros, it disappears, and we'll left this duct. We will not take into account kidneys, definitive kidneys, and we'll look only at these ducts. So let's look at the development of the reproductive tract. We'll left Wolffian or mesonephric duct, and it plays key role in the development of male reproductive system. So we'll show this duct in blue shade, and also a little bit later near this duct another duct appears. There are two ducts on two sides. They are called paramesonephric ducts. This duct is mesonephric duct or Wolffian duct, and also there are two paramesonephric ducts or Müller's ducts. So this is duct of Müller, this is duct of Wolffian and Müller's duct. And Müller's duct plays key role in the development of female reproductive tract. Here we can see this Müller's duct. And we will show this in red shade to distinguish between male and female ducts, so-called male and female. So male is mesonephric duct or Wolffian duct, and female it's paramesonephric or Müller's duct. And both ducts, note that both ducts, they are present in female and male organisms on the indifferent stage. So germs of female and male reproductive tracts are present in both in girls and boys on early stages of their embryogenesis. And here we can see it was only one Wolffian duct, and now there are Wolffian and Müller ducts. Two ducts are present. They are located near the developing gonads, near the genital ridges. And later, these paramesonephric ducts, Müller's ducts or female, they are fused and they form a V-like shape, and it's place of their fusion. And it will form uterus in female organism. It will be uterus and two fallopian tubes. But this structure is present also in boys. And here you can see final indifferent stage, final achievement. So here we can see female paramesonephric duct. Here we can see it common part, fused part. It will be uterus, body of uterus, and two fallopian tubes. And its male system, this mesonephric duct, which is opened into urinary bladder, and also ducts of Malpighian bodies, which were functioning like nephrons in embryo, they remain and they are growing to the gonads here. And let's look what will happen in female and male ways of development. So it's final achievement of the indifferent stage, and next stage it will be differentiated stage. In female organism, male duct, this mesonephric duct, Wolffian duct, it disappears. There is a special if the testosterone is not produced, this duct it disappears, and only paramesonephric duct remains. Here we can see this paramesonephric duct. And this place of those ducts, place of fusion, it is enlarged and the uterus develops. So it will be uterus, two fallopian tubes, and two ovaries, and its development of female reproductive system. And here we can see fusion of those ducts, formation of the uterus, fallopian tubes, and ovaries, and the development of this female reproductive tract. And ovaries, they achieve their final position, and here we can see the development of female reproductive tract. And here we can see fusion of those ducts and formation of the lumen of uterus and two fallopian tubes. It's development of this part. And there are different abnormalities of the uterus and vagina which are caused by abnormal development and fusion of the paramesonephric ducts. So different kinds of duplication of uterus may occur as a result of incomplete fusion of those ducts. So sometimes there are two vaginas and double uterus with one fallopian tube. Each has one fallopian tube. Sometimes they have septum between them. Sometimes there is one vagina but two uteruses, or B-horn uterus with incomplete fusion, or uterus with septum, or only one paramesonephric duct remains and another disappears, so uterus has only one fallopian tube, or one fallopian tube is obliterated, or these ducts may not be developed at all and only vagina will appear. So different kinds of abnormalities, they may occur on this stage of the development. It's result of incomplete or broken fusion of the paramesonephric duct. And here we can see uterus, its fallopian tube, its normal condition, and remains of the mesonephric duct, male, also may be present in female organism. Near the fallopian tube, there are apophoron and parophoron, bubble-like structures which are present in the ligaments which surround the fallopian tubes, and usually they are described as a normal condition, so they are remains of the male mesonephric duct. And sometimes they may undergo inflammation, so they can give signs, clinical signs, and different complaints from the patient. And near the vagina, this lowest part of the mesonephric duct remains as a Gardner cyst. So it also remains of the mesonephric duct. And let's look at the male way of development. So if there are testes, if Y chromosome is present, it results in the male way of development of gonads. So testes are developed and Sertoli cells appear. They begin to produce anti-Miller's hormone, which activates disappearance of the Miller's ducts. So anti-Miller's hormone activates these cells and they activate their apoptosis and these ducts disappear. And only male ducts are present now. Here we can see developing testes. This mesonephric duct, it will be ductus deferens, it will be ductus epididymitis, and they will be ductuli efferentes. And here we can see testicular cords. They will form convoluted seminiferous tubules, straight tubules, and rete testes. Own tubules of testes, they will develop from testicular cords. And seminiferous tract, beginning from the ductuli efferentes, ductus epididymitis, and ductus deferens, they develop from mesonephric duct and tubules of the Malpighian bodies, which were working like small nephrons in the embryogenesis. So here we can see that these structures, they remain in the male organism and play key role in the development of male reproductive tract. And here we can see development. It's embryonic testes. There are testis cords, rete testes, and those tubules, they remain here. There are epigenital and paragenital tubules and mesonephric duct, which is opened now into the urogenital sinus. Later it will be opened in the urinary bladder, and then it will be opened in the urethra. Later we will see what will happen. And also there are remains of the female paramesonephric duct in men. We know that some male structures, embryogenic structures, remain in female organism, and the same thing happens in male organism. So here we can see apex near the testis, apical part of the paramesonephric duct, it remains as an appendix testis near the testis. So it's appendix of epididymis, and near the fusion of the paramesonephric ducts, this part remains as utriculus prostaticus. It's small diverticulus of the urethra, which may be found in prostate. So it's so-called male uterus. It's very, very small diverticulus of the urethra, which is embryonic remains of female germs of uterus. So a germ of uterus was present in male organism, and it remains as very small diverticulus of urethra inside the prostate. It's called utriculus prostaticus. And also vas deferens, it forms seminal vesicles, and this will be ductus ejaculatorius. And here we can see mesonephric duct. It was separated from the ureter, so reproductive and urinary systems, they were separated here, and this duct goes deeper and it moves to the urethra, and now it's opened into urethra, not in the urinary bladder but in the urethra. And later testes, they descend, they are moving to the scrotum. We call this descent of testes, and testes finally they achieve their final placement. They are moving from the abdominal cavity to the scrotum. And also there is a processus vaginalis. It's the process of peritoneum which follows the testicles, and later it will form tunica vaginalis testis. It will be separated from peritoneum, but it has structure of serous membranes, so it's derivative of the peritoneum, which grows together with testicles and moves into the scrotum. And here we can see developing testis, its kidney, and testis moves deeper together with mesonephric duct and achieves the final placement in the scrotum later. And here we can see kidneys, testes, and here we can see ureters and mesonephric ducts. It will be ductus deferens, and those ducts are moving deeper. They are moving to urethra. And here we can see the ureters. They are moving upper. And we can see trigone region or triangle of the urinary bladder. It's place where different ducts are migrating, so it's place of embryonic changes and embryonic displacements, and it's known as a trigone region of the urinary bladder or triangle of the urinary bladder. And it hasn't submucosa, and mucosa is attached directly to the muscularis, so it hasn't folds. And ureters are growing upper and testes are moving lower. And let's look also at the development of the female reproductive tract. So we already know about development of uterus. And uterus now, this paramesonephric duct, common duct, is opened into urethra, and it's called synovaginal bulb. And later it will form vagina. It will be separated from the urethra, and it will form vagina. So here we can see its urogenital sinus, and it will be formed into the urinary bladder. It's paramesonephric duct female, and later it will be opened into urethra, and later it will be separated from urethra and fused with ectoderm. So female reproductive tract is separated from the urinary bladder, as well as urinary bladder was separated from the rectum. And here we can see final separation of these structures. And ectoderm is fused with vagina, and here this fold of mucosa where ectoderm is fused with vagina, it's known as hymen. Here we can see its fold of mucosa, which develops from ectoderm, particularly from this germ of vagina, and its place of embryonic fusion. So its paramesonephric duct was opened into the urinary bladder, embryonic urinary bladder or urogenital sinus. Later it moves to the urethra, and later is opened individually and fused with ectoderm. So students, we have discussed stages of development of reproductive systems. There are indifferent stages and differentiated stages, and in different stage when we can distinguish boy and girl, and differentiated stage is own way of development, female and male. And also there are development of gonads. They develop first, and under influence of hormones which are produced by gonads, reproductive tract has female or male way of development. So students, that's all, and thank you for your attention.